Hi all, I am Callie, and today I'll be talking to you about the structure and life cycle of influenza viruses. The influenza virus, or the flu as we sometimes call it, is actually a part of a family of viruses called Orthomyxoviridae. It's a family. This encompasses several other viruses in addition to the four influenza viruses. A, B, and C can infect humans, and the fourth, D, is primarily known to infect cattle and don't have a known human host. You can see this nice phylogenetic family tree that I have here with nodes denoting branch points with the last common ancestor and point of genetic divergence, with branch length denoting time of divergence. You can also see that some of the A strains, which are the most common, emerged earlier than the two B lineages. C is the least common type of influenza and not known to cause pandemics. In fact, the A lineage is responsible for pandemic flus, just the A lineage. Influenza viruses are negative sense ssRNA viruses, meaning that the negative sense 3' to 5' viral RNA is complementary to the viral mRNA and thus, or the viral RNA, or mRNA. Therefore, a positive sense RNA must be produced by an RNA-dependent RNA polymerase from it prior to translation. As I mentioned, influenza viruses are largely divided among three main types, A, B, and C, that infect humans at least. Influenza A viruses are divided into subtypes. Hemagglutinin, or HA, and neuroaminidase, NA, are the major viral glycoproteins that are detected by antibodies and that define the subtype of the virus for type A influenzas. There are 18 HA subtypes and 11 HN subtypes, respectively. This is how they get their naming scheme. So when you hear H1N1, that's denoting their hemagglutinin type and their neuroaminidase type. And that's how they come up with type A flu nomenclature. Type B influenza viruses are not divided into subtypes, but rather one of two distinct lineages. The lineages, the B Yamagata 1688-like and the B Victoria 287-like viruses, are named based upon the properties of their hemagglutinin. Both A and B type flus can further be subdivided into clades and subclades, with the exception of the Yamagata type B lineage, which can be subdivided into clades, but no subclades exist. These distinctions rely on sequence differences between their HA genes. C is found in humans, but less commonly, and it's rarely responsible for pandemics. Actually, it's not responsible for pandemics, and I'll give you the reason for this later. But type B influenzas have a more limited host range than type A influenzas, and this limited host range, strictly seals in humans thus far, is thought to be responsible for its limited pandemic spread. Although its number of zoonotic hosts and comparatively low mutation and evolution rate to type A flus make it a poor candidate for initiation and spread of pandemics, type B flu lineages are responsible for seasonal flu epidemics that can be associated with high morbidity and mortality rates. So something to keep in mind is that the structure of the influenza virus is somewhat variable, and I feel like the size is interesting because when you think about it, a range of 80 to 120 nanometers I feel like that's a pretty wide range for a virus. But so A, B, and C are envelope viruses. Variant particle size is usually somewhere, as I mentioned, between 80 and 120 nanometers. So the size, as I said, ranges quite a bit, and they're usually spheroid or ovoid in shape, capsid shape, and filamentous forms of the virus can occur as well. But these are more common among some influenza strains than others. Enveloped influenza A viruses have three membrane proteins, the hemagglutinin, the neuroaminidase, the HA or the NA, and the M1 and M2 matrix proteins below the lipid bilayer. The M1 and the M2 are just matrix protein 1 and matrix protein 2, and the M2 is the matrix protein below the bilayer, I guess, to clarify. And that lipid bilayer is derived from the host plasma membrane. I know this was a question we had on one of our discussion boards a few weeks ago. They also have a ribonucleoprotein core, consisting of 8 viral RNA segments and 3 proteins, the PA, the PB, and the PB2, the PB1 and PB2, and the nuclear export protein, or the NEP protein, for influenza A. And I will explain in more detail later, but the PA, PB1, and PB2 are polymerase subunits. Influenza B variants have 4 proteins in the envelope, the HA, the NA, the NV, and the BM2. Remember how I said that variants are usually ovoid or spheroid in shape, but there's some variability? Influenza C viruses have hexagonal surface shapes like the influenza A and B viruses. The core of the influenza C viruses consists of a ribonucleoprotein made up of viral RNA and 4 proteins. The M1 protein lies just below the membrane, as in influenza A and B variants. A minor viral envelope protein is CM2, which functions as an ion channel. The major influenza C virus envelope glycoprotein is called HEF, for hemagglutinin esterase fusion, because it has the functions of both the HA and the NA, which I'll explain. But therefore, the influenza variant for type C is 7 RNA segments and not 8 RNA segments, like influenza A and B viruses. Or rather, gene segments, I guess you could say. The HA protein precursor for influenza A life cycles, that hemagglutinin I talked to you about, forms these spikes on the lipid membrane of the virus, and they bind to the sialic acid residue found on the surface of the host cell's membrane. And then when that happens, receptor-mediated endocytosis occurs, and the virus will enter the host cell in an endosome. And the endosome, as we've discussed, has a really low pH, around 5 or 6, and that's super important. That actually triggers the fusion of the viral and endosomal membranes. There's a precursor form of the hemagglutinin, and that low pH induces a conformational change in that precursor, and it exposes one of the subunits that acts as a fusion peptide, or has a fusion peptide, and this fusion peptide inserts itself into the endosomal membrane, bringing both the viral and endosomal membranes in contact with each other. And then that acidic environment isn't just important for that conformational change of that precursor, but it opens up that M2 ion channel that I talked about, and it forms this little pore, it's a tetramer, and when it opens, hydrogen ions flow in and acidify the viral core, and that releases actually the viral ribonucleoprotein from the M1, and then the viral ribonucleoprotein is free to enter the host cell's cytoplasm, but that acidification step really is key. After being released into the cytoplasm, the viral ribonucleoprotein will enter the nucleus, and so the viral proteins that make up that viral ribonucleoprotein are the NP, or the nucleoprotein, the PA, or the polymerase acidic, and polymerase basic 1 and basic 2 proteins. And then all 4 are necessary for transcription, so this is super important. All of these proteins have known NLSs, or nuclear localization signals, that combine to cellular nuclear import machinery and then enter the nucleus. Influenza A uses the important alpha, important beta nuclear import pathway to gain entry into the host cell nucleoplasm. And then negative sense viral RNA needs to be converted into positive sense RNA, as I mentioned earlier, to serve as a template for the production of viral RNAs. Replication of the genome does not require a primer, but instead the viral RNA-dependent RNA polymerase initiates RNA synthesis internally on the viral RNA. After transcription, the viral ribonucleoproteins exit the nucleus through the nuclear pores via a mechanism that isn't fully understood. It's hypothesized that M1 might bind to the negative sense viral ribonucleoproteins, as well as binding to the nuclear export protein. And then the nuclear export protein will bind to that CRM1 I mentioned, and then that chain allows the viral ribonucleoproteins to be exported from the nucleus. From there, viral protein transcription occurs on cytosolic ribosomes for the using host machinery. for all viral proteins except the membrane proteins, the HA, the NA, and the M2, which occur on ribosomes localized to the endoplasmic reticulum, as you probably remember from your cell biology courses. From there, and actually that has an apical sort of face to it, that it's has apical polarity or directionality, where they're inserted into the membrane. And that becomes important during your release, but from there, the nuclear protein and the PB1, PB2, and the PA polymerase subunits are targeted back to the nucleus. At that point, the viral mRNA transcription and viral RNA replication can occur. Since influenza is an envelope virus, it uses the host cell's plasma membrane to form the viral particles that leave the cell and go into infect neighboring cells. The HA, the NA, and the M2 are transported to the apical plasma membrane, like I just mentioned earlier, that sidedness to it, the directionality, and virus particles bud from the apical side of those polarized cells, and there used to be this thought that it was maybe random packaging into vesicles, but there's evidence that indicates that it's specific, and it depends on the virus having signals present in viral segments dictating which segments are to be packaged into the virions, making it specific. And like I said, there's some evidence for it. One of the most important steps, though, that must occur before the newly made viral particle can leave the plasma membrane, and that's that the sialic acid residue that they originally bind to enter the cell from has to be cleaved from glycoproteins and glycolipids in the membrane. So the neuroaminidase removes these sialic acids, and without this process, the viral particle wouldn't be released from the plasma membrane. An interesting fact is that the M2 protein is the target of the amantadine class of anti-influenza drugs, which block ion channel activity and prevent viral encoding. So although influenza B has many of the same proteins influenza A does, they have two extra proteins, the NB and the BM2. The BM2 protein of the influenza B virus functions as an ion channel, which is suggested to be important for viral encoding in endosomes of the virus-infected cells. Attachment occurs in the same way as A, with HA, the hemagglutinin, attaching to the sialic acid on the surface of the host cell. Endocytosis occurs for B-type influenzas as well as in A, and endosomal acidity contributes to viral encoding. Just like in type A influenza, though pH triggers HA conformational change that exposes a fusion peptide that allows the viral envelope to merge with the endosomal membrane. But unlike influenza A and influenza B, it doesn't have an M2 channel, but rather the aforementioned BM2. Acidification of the endosome is thought to again be a crucial process that allows disruption of those protein-protein interactions within the endosome, and it facilitates release of viral RMPs from the viral matrix and into the cytoplasm. Nuclear localization, transcription, synthesis, and the viral budding and release appear to be more or less the same between influenza A and B, with the exception of the differences in DNA coding due to the BM2 and M2 and then NB differences. Influenza virus budding occurs at the cell membrane, probably initiated by an accumulation of the M1 matrix protein at the cytoplasmic side of the lipid bilayer. When budding's complete, HA spikes continue to bind the virions to the sialic acid on the cell surface until the virus particles are actively released by the sialidase activity of the neuroaminidase protein that I discussed on the previous slide. It possesses an activity that basically is enzymatic and cleaves the terminal sialic acid residues from the cell surface, the glycoproteins and the gangliosides I talked to you about. And then it releases the progeny virus from the host cell. This is common to A, B, and C. Lastly, C, the major difference is that the HEF protein I talked to you about is what mediates attachment to sialic acid in influenza C, meaning its attachment stage is different than what we see in A and B. It appears to be the major distinctions between C, A, and B are the HEF protein, and then the existence of 7 gene segments instead of 8 for C, or in the case of B, that BM2 protein. So that is my presentation, and these are my sources for anybody that is interested. I have the image sources listed. Thank you so much for letting me give you this presentation.